Okay, hello, good evening. Welcome to today's session. First, I'm going to go over a discussion for the topic that was discussed in the last class. As part of the original flyer that we had discussed, whereby we were covering the entire topic of advanced concepts in cancer biology and precision medicine, uh, our major focus has mainly been on understanding cancer biology, the basics of it, following it up with the understanding of the signaling pathways which help in the onset and progression of the disease. And furthermore, we moved towards the detection techniques. Majorly, we focused on the traditional methods that were more invasive in nature, that involved surgical procedures. And then we moved on to techniques that are more non-invasive in nature, which means that they included sampling of some part of that patient's body. Majorly, these were body fluids like or the saliva or urine, which is much less invasive and easier procedure, much more practically feasible to obtain that sample without having to subject that patient to a lot of unnecessary trauma, and also simultaneously ensure very accurate results using those easy to access samples. Then we discussed in the last class, we went into discussions about some advanced techniques in cancer therapeutics. We understood why a certain class of proteins or certain targets in cancer are usually considered to be undruggable. Majorly, these are the transcription factor family. These include the transcription factor family, which are usually considered to be undruggable. However, at the same time, they are considered to be very key drivers of the entire oncogenic process, which means that their involvement in the onset and the progression of tumorigenesis is usually their contribution towards that process is very, very significant. And therefore, they are very good targets. However, their druggability is something which is very severely compromised. So, this remains a tug of war which persists till date. And then we finally understood a little bit about precision medicine. And as I had mentioned in the previous class that I was going to discuss, I was going to move a little bit away from this topic because there are a few more discussions that we are going to complete today. So we'll move on towards more discussions about the um, model systems which are required in context of cancer. And in the next class, we will resume some of the remaining discussions, which are a little more detail about precision medicine, understanding some key terminologies in cancer statistics, and finally, we'll talk about drug resistance. So I wanted to break the monotony a little, so I diverted from this flowchart, and today we are going to discuss cancer models instead. Basically, the model systems which are used for cancer research or in oncology research. So that will be the focus for today. Now, before we go into that topic, I'll just quickly brush over whatever was discussed in the last class. Majorly the focus was on understanding the current approaches which are taken towards cancer therapeutics, diverting away from the traditional route of going, through radiation and chemotherapy, and going for the approach where you just look at anti-proliferative drugs like taxanes, like we saw, vincristine, vinblastine, etc., which all interrupt the cell cycle at some point or the other. Now, we discussed the shortcomings of those chemotherapeutic drugs. While they are very rarely, they are actually used even today for more advanced patients. Like, for example, if a patient has progressed to a stage where the default prescribed route for treatment, like for example, take if you talk about say a stage 4 prostate cancer patient. So that patient is usually somebody under normal circumstances, it is somebody who has undergone a lot of treatment previously, and therefore at this point they have developed so much resistance to all of the ideally prescribed drugs like AR antagonists, androgen deprivation therapy, PARP inhibition. All of these strategies have already been used for that patient, and it has not worked in their case. That after that, eventually people progress. Even eventually, the doctors they prescribe something which is not very ideal, but they do go for a prescription of Taxols and vincristine, vinblastine, etc. Because in that case, it's more about the risks are something the patient can afford at that point of time. So it's about so, like what I wanted to explain was that while they are more outdated methods of treatment, it's not that they've become completely irrelevant. They are till date in the market, but they're not the default route of treatment. So here we had started off with a discussion about how we have advanced from that um, previous approach towards what is recommended nowadays. So as you can see, first we talked about chemotherapeutic breakthroughs very early on in the pre-1990s, basically. Then targeted cancer therapy using rituximab was discussed, trastuzumab, which we have already discussed a long time back when we talked about the signaling pathways relevant for cancer, imatinib, gefitinib, sorafenib, all of these we have discussed at some point of time when we talked about using RTK inhibitors, receptor tyrosine kinase inhibitors for treatment of cancer patients. So again, to reiterate this, we had discussed targeted therapy separately because it was related to what we were talking about at that point of time, which was about the signaling pathways, which help in development of cancer and progression of cancer. So, in that context, we had discussed it. But please keep in mind that targeted therapy is not the most traditional route. It is something which has emerged over time, and now it is the taken approach, and therefore you should definitely consider it to be an advanced method of cancer treatment, and therefore cancer therapy. When we talk about advanced cancer therapy, targeted therapy is something you're going to see almost throughout here. Like trastuzumab, we had discussed targeting the HER2 new receptor. All of these we have come across at some point of time during the course. Um, what I had also iterated here is that whenever you look at the name of a drug, just keep in mind that if you say MAB at the end of the name, then it means it is a monoclonal antibody, which is prescribed for that treatment, and therefore um, it falls under the purview of immunotherapy because it involves activating the immune system to eliminate or to at least sensitize the immune system against that tumor which is persisting in that patient. So yeah, this was just a timeline based perspective, so you can go over it in your time. I'll share the slides either by today or latest by tomorrow, the slides and recording both for the last class and today's class. So then we moved into the discussion about small molecule inhibitors, so which by the virtue of their very name mean that they are very small molecules. The molecular weight is less, and they are inhibitors, which means that they do not. Yeah, here I had also stressed that you need to understand the fine differences between an inhibitor and a degrader. So an inhibitor is something which just binds to its target protein, and it stops it from functioning further. Whereas a degrader will be something which will tag that target protein and will take it for removal, take it for clearance or proteasomal degradation. So these terms should not be used loosely, should be very careful with their usage. And in this case, when we talk about SMIs, they are small molecular inhibitors. So what they essentially did was they blocked some specific molecules by interaction. Typically, those were proteins involved in cell signaling pathways or metabolism or normal gene expression, and they also bound to target molecules which helped in disruption of their function and therefore interrupted the development of cancer eventually. Uh, and the mechanism which is followed by small molecule inhibitors is that they bind to the pocket which is present on the surface of their target proteins. And by this pocket, we basically mean that the 3D structure or the final quaternary structure of the protein, if you've studied basic structural biology, you would know. So the tertiary or quaternary structure of the protein, if it has a some sort of a socket or like a region which is sort of protruding inside, then um, it allows a good scaffold or a good binding site for the docking of any inhibitor, and that allows it to stay bound once it has bound already. Basically, if you study terms like k on k off, which is which is the affinity constant, which lets you know how strong the interaction between two entities can be. So if you have a socket, if you have a structural support for the binding of one inhibitor to one particular protein, then that results in a much stronger interaction and the inhibition would be much more efficient. So this is the mechanism by which SMIs work, that because they are so small in size, they're able to fit into these tiny pockets and they're able to interrupt the functionality of the normal protein, which aids tumorigenesis or which aids cancer progression. So they are very efficient at their job, and because of by virtue of their small size, they are able to have a very wide range of targets, which is actually an advantage over antibodies because antibodies are obviously more bulky in size. So like for example, your antibody would at least have to be a few kilodaltons, like at least 50 kilodaltons is the light chain, and heavy chain is somewhere around 120, 110 or 120 kda. So effectively, the net molecule is actually quite heavy. It's around 200 kilodaltons. So that's a bigger protein, and not just the molecular weight, but also the architecture or the physiological structure of the antibody is such, you know, that it has that Y structure, right? So that is not always able to enter into all of these pockets, and so it poses steric hindrance. So SMIs have an advantage over that. Furthermore, they're able to penetrate the blood-brain barrier, and therefore they're very helpful for lesions within the brain as well, which antibodies are obviously restricted. They cannot pass through the blood-brain barrier, and therefore SMIs have a lot more potential. So we had looked here. You can see at a lot of examples where these different types of SMIs can bind, like for example the RTKs, which we've already discussed in a lot of detail, inhibitors of the RAS-RAF-MEK-ERK signaling pathway, PI3K-AKT pathway, inhibitors of the cell cycle, inhibitors of um, nuclear processes like the central dogma of molecular biology, like transcription, translation, etc., inhibitors of mitochondria mediated death signaling, which is the apoptotic signaling. So all of these key procedures, they are interrupted by small molecule inhibitors, and therefore they were very promising in their capability in context of cancer therapeutics. So then we talked about this group of proteins which are normally considered to be undruggable, and somebody had asked why exactly are they considered undruggable. um, the basic reason for that is because transcription factors, which is the group we are referring to here, uh, in most cases, first of all, they're quite small in size. Second of all, under most circumstances, transcription factor structure is such that it does not have a lot of pockets. Think about it very logically. What is the job of a transcription factor? It is supposed to bind to DNA because that is what will allow it to recognize its response element and trigger the transcription of the downstream gene. Like, for example, AR will bind to the AR response element, trigger the transcription of the AR regulated gene. That's how it works. Apart from that, the surface of AR has to interact with all of the regulators of the entire transcriptional process, like epigenetic modifiers, chromatin remodelers, all of these other proteins which are involved in the circuitry of transcription. So those proteins do not necessarily need a pocket to be present, right? Any protein which has a pocket is usually something which will have a ligand. Like, for example, receptors are very good ways of targeting because they always will have a, well, always is a strong statement to make, but they almost always have a very clear pocket where the ligand can come and bind, right? That is not feasible in context of these kind of proteins, like these transcription factor group of family. And therefore, while they are very promising targets when you learn about cancer, why very promising? That is because they are, if you look at any paper where they have studied the genomic landscape of any cancer, you will almost always find that the most predominant class is either receptor tyrosine kinases or it is the class of transcription factors that they are driving cancer in patients. Like almost every patient is going to have some or other mutation in either of these two pathways. Now, while RTKs are very good in their druggability context, transcription factors are very horrible. So while it is coming up as a wonderful target, it's not very easy to drug. It's not very easy to target, inhibit, or remove from the patient's cancer cell. And therefore, a lot of people could not really receive treatment for the condition that they had. So therefore, for a long time, this has remained a challenge. A lot of strategies have come up. Like you can find entire review papers and books on this, whereby people have studied transcription factors over decades and tried to target it, and they've used many different approaches. None of them have been very foolproof, but they have tried a lot of different mechanisms. So, one of the mechanisms that has been tried and that appears to be a wonderful discovery um, that seems very promising is the use of covalent modulation of a small molecule inhibitor. So covalent modulation basically means you are modifying the structure of the small molecule inhibitor covalently, and as a result it is able to form interactions, chemical bonds or interactions with some part of that protein. In this case, the example protein is sotorasib, not protein but the small molecule inhibitor is sotorasib, which has modifications that only and only bind to the mutant version of the KRAS protein. And the KRAS protein, like I explained previously, is one of the key mediators of the RAS-RAF-MEK-ERK signaling pathway, the MAP kinase signaling pathway, which is very crucial for cell growth, cancer cell growth, and sustenance. So, what this inhibitor does is, because of the unique modification that it has, the covalent modification that it is only able to go and bind to this mutant residue and it will not bind to the normal KRAS protein. So, that gives it a very high specificity. And because it has now bound, what it effectively does is it compromises the functionality of this mutant protein, patients who have this G12C mutant protein. And therefore, what it does is it locks this KRAS in an inactive state and it prevents it from binding GDP. Therefore, all the downstream pathways are shut off. And instead, only the normal regulation, which is mediated by the normal protein, can only take place. So, this discovery was a huge revolution in the field of not just cancer biology, in the field of drug development, drug discovery. And so, that was one of the most remarkable discoveries. So, this is just one of the approaches. But apart from that, there are other alternative strategies that have also been taken up, like we talked about allosteric inhibition. So basically, what you do is you use an inhibitor, like in this case, the example as asciminib. What it does, it binds to a different site from the ligand binding site on your protein of interest, which is called as an allosteric site. Now, I had mentioned in the previous class that if you've studied enzymology or just enzyme biology in general, you would know what an allosteric site is. It is basically a site which, upon binding to the ligand, modifies the ligand. Upon binding to the inhibitor or the regulator, it modifies the ligand binding site in some way. It undergoes some sort of structural modification. So, in this case, the example given was BCR-ABL, which is a major driver of chronic myelogenous leukemia. And when asciminib, the inhibitor, the allosteric inhibitor, binds to this protein, it locks this protein in its inactive conformation, and therefore, patients with T315I mutation, they are now able to survive much better because this mutant protein is no longer able to function, and so the patient eventually undergoes remission. Then we also looked at the example of cobimetinib, which is an oral MEK1 inhibitor, also used for melanoma research. So MEK1, again, the stress on the fact that this RAS-RAF-MEK or signaling pathway is a wonderful strategy and is very commonly used across different types of cancers. So this cobimetinib drug, it inhibits MEK1 and 2, and therefore it shuts off or it basically disrupts the pathway at this step. So further phosphorylation cannot take place, and so the growth signaling sustenance loop is basically lost. So cells will eventually, if they are very dependent on this pathway, they will eventually die. And so these drugs, they seem very prominent and very prominent in the sense that they are being explored extensively, not just in research lab capacity, but also they are being subjected to clinical trials to see how well they can be approved. And so they're very interesting targets, and let's see how much of it can actually be used in patient settings. Then we looked at the next strategy. So this one moves into the next set of drugs. By the next set, I mean we had first talked about inhibitors. Then I had mentioned that there's a difference between these inhibitors and another class of drugs known as the degraders. So these degraders involve, again, keep in mind, I have not mentioned all the different types of protein degraders that can be used. I've only talked about PROTACs here. There are different types of degraders as well. You again will find entire review articles on just this topic by itself. So if it is interesting to you, please go ahead and have a look at it. But what I had mentioned here are PROTAC degraders. So PROTAC is proteolysis targeting chimera. So it basically has two motifs. One which has the POI ligand, which has a ligand or a bait moiety, which is able to attract our protein of interest and allow it to bind very tightly. On the other hand, it is attached with a linker, which is connected to the other end at the other end with a E3 ubiquitin ligase ligand. So as you can understand from the as you can understand from the name, E3 ubiquitin ligase is part of the proteasomal machinery system, and therefore when its ligand is able to also eventually bind to our protein of interest, what it effectively does is it targets this protein for degradation and allows all resources to be recycled as well. And so more and more protein will eventually get degraded. So if you have your some mutant protein and you have somehow designed a PROTAC against it, then the PROTAC will go and call for or will tag all of that protein for degradation, and so only normal protein will remain, is ideally supposed to remain functional within the cell. Why ideally is because, as you can understand, these are not very foolproof techniques. There are a lot of uh, off-target effects because specificity is something that people have been, really struggling with, not just in context of PROTACs, but for any drug for that matter. And also, sometimes it may so happen that your degrader is very efficient at removing a certain protein, but its efficiency is so unnecessarily high that it is getting rid of your normal protein as well. Now in a lot of situations that can actually make things worse, and then normal protein might not be available for the cell to utilize, and it might be survival essential, so it might compromise the health of normal cells to a much larger extent. So these are ifs and buts which are associated with it, but basically this is the idea of a PROTAC. So it is a proteolysis, it is a chimera. Chimeric molecule means it's a fusion of two different types of DNA, which in this case is the two or more types of DNA, which in this case is the protein of interest ligand linked to a linker, which is again linked to another ligand. So this chimera helps in protein degradation basically. Then here a couple of examples were mentioned. So you can certainly go over that in your own time. Yeah, I think I'll rush a little bit through this. Um, then we had discussed antibody drug conjugates, whereby you use the specificity and the unique nature of antibodies binding to antigen to our advantage. We tagged it with some sort of toxin, which will allow very specific, very targeted delivery of our toxins of interest to our to a certain subpopulation of cells, which will be decided based on what type of antibody we are using, and then it eventually leads to killing of that cell, of that affected cancer cell. And this is often referred to as a biological missile, and the antibody serves as the guidance system, the toxin acts as the warhead. So this helps you in very precise removal of cells from the cancer cells from the body. Here, some more detail was provided. Please look at that. I'll brush through it because it is something you can do at your own time. Then we understood a little bit about precision medicine and how it has shifted from traditional approaches under normal circumstances. So we talked about the fact that precision medicine, the name given to it, arises because of it caters to the makeup, the genetic makeup of the patient himself and the cancer within the patient himself, instead of using a very generic idea to treat a certain patient. So it takes information about the patient for the patient and for more targeted, more accurate treatment strategies. So this is an innovative approach which uses an individual's genomic, environmental, and lifestyle information to guide decisions related to their medical management. The goal of precision medicine is to provide a more precise approach for the prevention, diagnosis, and treatment of that particular person suffering from the disease. So it has shifted from the idea that you can have just one treatment and that's going to work for everybody. And from that, it has moved into gathering information about that patient, about their lifestyle, their genetic background, their influence by other factors, and taken that all into truly analyze the spread of cancer within a certain population, or might be within even a family. It's basically it's for the people. It's more, so to say, democratic in nature. So that's the basic idea. However, the nitty-gritties of what precision medicine by itself is, that is going to be a very long discussion. So I think we did not go into it eventually. Oh, sorry. We did not go into it in too much detail, but I will be introducing a little bit more about precision medicine and a couple of other advances in cancer therapy, like subtypes, in the next class. So, like, I would want to discuss about epigenetic drugs. I would also want to go into a little more detail about immunotherapy and how it is being harnessed in context of cancer research, all of those things. Finally, we understood a little bit about CAR T cell therapy. If you're already familiar with it, then well and good, not much to stress about. But if not, you can just go over the slide. It's very simple. It basically involves developing some immune cells which are specific to your target cells of interest, and in their accurate and very precise removal from the patient's body. And it involves genetic engineering, which means that, like within the lab, you are designing the DNA, which is going to produce your protein of interest, so that it helps with the uh, removal as in the so that it helps in eliciting a proper immune reaction for the same. Sorry. So yeah, again, this I have discussed this extensively in the last class. We discussed about all of the different types of sorry, sorry. So we discussed about all of the different types. By types, I mean all of the progress that has been made in context of CAR T cell generation. So yeah, here you can see how modifications have been made over time from the first generation to second, third, and fourth, what all has been added, how there has been addition of co-stimulatory domains and stimulatory domains to make it more specific, how the single chain variable fragment has undergone modification. So all of this we discussed in the last class. So you can go through it in a little more detail. It should be fairly simple to figure out. If not, of course, we can have a discussion on the same. So we had ended the last class on this note. If there are any queries, please unmute and let me know, and then we'll move on to the discussion for today's class quickly. Okay, if not, we're going to move ahead. So, switch this on. Yeah. So today we are, like I previously mentioned, we are going to look at the ways by which cancer is studied in context of academic research or medical research, or just um, like how you exactly arrive at a conclusion that this is the way cancer is progressing and this is the way you're going to design a drug and this drug is effective, not effective, all of those things. So you might be familiar with these concepts already, if you've worked in a lab or if you've studied like cell culture, etc., all of these before. So if not, I wanted this to be a part of the topic so that anybody who is not familiar with it does get to know. So just go along with it. So starting off with the fact, how do we study cancer? So cancer models, yeah, of course, first you need to have this idea in mind that for figuring out how anything happens in nature, you always need a model, whatever disease it may be. Now, there are different types of model systems. You might already have studied these in your bachelor's or your master's if you've had like animal physiology or topics like these. So basically, you will always need to use a different system for studying any disease, because humans, of course, you cannot use for your just for studying a disease. You can test the feasibility of a certain approach, or you can test the like, how do I explain? You can use some part of actual human beings for your research, but you cannot use them as guinea pigs. Let's just put it like that. There's a lot of bioethical clearance that goes into approvals like this. So therefore, we shift to model systems where we will first figure out the disease. We'll check all our boxes, and then we move to humans, right? So when we talk about cancer, then basically what you need to have some model system which mimics what happens in an actual individual who is suffering from the cancer, and for that you use different types of models in that context. So two major domains are being shown here. Either you have experimental systems or you have computational models. So experimental systems for studying human cancer include, of course, cancer cell lines, which is something you almost everybody would be familiar with if you've worked in this regard. And what we will be eventually moving towards are the 3D organoids. What are organoids? We'll look at all of that, don't worry. And you also use other living organisms, like, for example, Drosophila, zebrafish, genetically engineered mice, GMMs, pigs, patient-derived xenografts, computational cancer models falls in the second domain. So you either use some part of a human being, by that we mean cell lines and organoids, or you use a different animal completely, where you can use all of these different approaches, like I just mentioned. So, um, what is the like if you had to make a comparison between all of these strategies, how would you do so? So for that, just look at this flowchart on the left. So if you look at in vitro models, including cell lines and organoids, you see that they have genomic alterations. Now the question is, you might think that why would you want that? Why would you want genetic alterations? That is because, like I just mentioned, you want to mimic the true condition in a patient. So you need to have some system within the lab which already has the conditions which you normally see in an individual. Let's take some examples. Like we just introduced a while ago, that KRAS G12C mutation is something very commonly seen in lung cancer patients. So if you were to study lung cancer in your lab, and specifically you want to study KRAS in lung cancer in your lab, how would you do so? You would need some sort of a model which already carries that mutation, and you would have a normal lung cell line model also for comparison between the two. So normal versus mutant is how you will make the comparison for any experiment within your lab. Say you want to characterize the addition of a gene, the removal of a gene, over expression of a certain gene, temporary overexpression of a certain gene, some drug treatment, a number of things can be compared. So for that purpose, you would need these in vitro models which carry these specific genomic alterations. Secondly, they have low success rate. That is the very sad part out of this whole thing, that while it looks quite idealistic because their maintenance cost is very low, in a lot of cases their maintenance cost is very low and less extensive in that regard. Their rate of establishment is not as high as you would think, especially certain cancers. Establishing cell lines by itself is so challenging. Even forget about organoids, that's way more tough. Third, there is no cell to cell interaction which can be studied here. There's possibility of cross contamination, and it is um, somewhat costly to culture. Like, for example, cell lines, even though they're not that expensive, every single component that goes into establishment of organoids is super duper expensive. And coming with a very low rate of success, it's not really the most idealistic situation, but that's just how it works. Apart from that, the in vivo models, if you are not clear about it, in vitro basically means something you are able to study in the lab, which means that you are studying something out of a system. So you're studying something in cells, or you're studying something in a population of cells. Versus in vivo means that you are studying something inside a system. Either it is inside a mouse, or it is inside a rabbit, it is inside chimpanzees, whatever is your model system. When you are studying the function of a particular gene, or you are studying a certain process within a whole organism, that is an in vivo study, versus something which is done with only parts of an individual organism, that is an in vitro study. So I hope that is like clear for you guys. And of course, examples will help clear up the difference between the two even better. So here you can see that under the in vivo models, you have GEMMs, genetically which are modified or genetically engineered in some way. The question is, why would you do so? That's because sometimes when you want to like I explained, the same mutation, the let's take that example. If you want to study that exact same mutation and see how it affects the development of a mouse. Of course, that is a different context. Here we're talking about a mutation which sets on or which emerges much later. If you were, say, for example, trying to study um, some other mutation which has a major role to play in development as well, then in that case, you would want to you actually want that particular mutation to be present in every cell within the animal's body, because that is what will help you truly understand what is the impact of such a germline mutation. And for that case, you would have to genetically engineer the mouse. So in other contexts also, transgenic mice are generated when you want to study the temporary expression of a certain gene. But yeah, in context of cancer, this is not always the route to be taken. Sometimes just having an immunocompromised mice is having mouse which are immunocompromised is good enough. So we'll discuss a little bit more about this later on about immunodeficient mice. But yeah, so these GEMMs, then PDX. PDX stands for patient-derived xenograft. Zebrafish, which are very commonly used for studying a lot of diseases, a lot of conditions, and even also they're very extensively used in ecological sciences as well. Then you have oncopigs and you have Drosophila, very commonly used for this purpose. Um, what are the prerequisites in this context? They have or they require immunodeficient hosts. Why? Because if you have a very active immune system, it is eventually going to, somehow kill the cells, and therefore you need a system where immune system is either shut down or is weak and is non-functional in the long run. Second, long tumor latency, which means that the tumor should be able to sustain for a definite period of time and shouldn't get immediately cleared off. Then you have a key disadvantage, being the fact that it is technically quite challenging. Not each and every person knows how to handle like a um, doesn't know how to breed Drosophila, doesn't know how to maintain mice, for example, might not know how to handle zebrafish or a genetically modified mouse. So all of that is practically quite challenging. Then it might be expensive to model multiple mutations. It's a lot cheaper to model if you have, say, 30 mutations. It's much easier to model 30 mutations in cell lines versus in a complete animal. And of course, it is ethically also much more questionable if you have to sacrifice 30 into X animals because you want to characterize the role of each gene at least, the role of that gene in at least three models. So it's just going to be very unnecessarily resource heavy, and so that's not the way to actually go ahead with the study. So what you effectively need are both opportunities. You should have access to cell lines, and you should also have access to in vivo models. um yeah, then the invasive method for drug administration might hinder the absorbance of the drug and the metabolism of the drug in the long run. What is mentioned here, I'm not really, it's not the focus of our study, but basically for your own knowledge, you can, look into it, which is about computational cancer models, which basically means you are using some sort of an in silico analysis to characterize a certain type of cancer. So you study the genome, the transcriptome, proteome, and metabolome for this purpose. You need to have specific mutations which are related to the drug response. You need to have, you should be able to identify potential novel biomarkers, drivers, and therapeutic targets for that particular disease. Sorry, I have a bad throat. Um, then you need to have modeling for how toxic a particular drug might be, etc. So overall, you use all of these information for modeling the response of a particular situation in silico. Of course, none of this information is relevant until validated in an actual model in a living system. So this is just preliminary analysis. Validation always comes from experiments. So taken together, experimental systems for studying human cancer include cell lines as well as 3D model organoids and organisms like I just mentioned. They help us understand the genetic pathways, the pathobiology of the disease, and it helps us understand the disease a lot better. So that is the basic conclusion. Here you can see a sort of comparison between all of the most commonly used types of cancer research models. So you have, okay, just for now, just have a look at it. We'll go into more details about these in the upcoming slides. But for now, just understand all of the different subtypes. So first and foremost, you have the 2D cell culture, most normally done in the lab. Then you have the Caenorhabditis elegans or C. elegans, a very, very common model system used for disease biology in general. However, its usage in cancer might be limited. But if you have studied key experiments, like for example, if you've studied siRNA, miRNA mediated silencing, all of these pathways, you would know that there are such huge contributions from organisms like C. elegans and the research that is done in them. Then you, of course, have Drosophila melanogaster, which is fruit fly, another very common model used. Actually, it's more commonly used for neurobiological diseases than for cancer because there's a lot of similarity between humans and Drosophila. But yeah, there is some usage in cancer as well. And there are some wonderful papers on it. You can check it out if it is interesting to you. Then you have fish models for this study for Daniel Rerio. Example here is Daniel Rerio. Now, you need to keep in mind that fish are actually more relevant in context of studying regeneration rather than studying cancer, while the two processes are effectively intertwined in the long run. But yeah, that's the general application that it is commonly used for. Then you have mus musculus, which basically means your regular mice, which are very commonly used. Then you have PDX, which are patient-derived xenografts, which have been implanted here, like you see, they have been implanted here in mice. And finally, you have human organoids, which seems to be extremely promising if you just look at all of the different characteristic features. So if you look at the comparison, you would see that there's a difference between the ease with which this can actually be, uh, the system can actually be established. Like for example, cell culture, 2D cell culture can sometimes be difficult to set up, but once set up, it's very easy to um, sustain. The ease of maintenance is different. Recapitulation of developmental biology, while it is almost completely lost in cell culture, it is nearly, yeah, it's almost retained in every other model system except for PDX. Duration of experiments can be quite different. Then you have, yeah, Ankit, go ahead.
And like in when doing this in vivo experiments,
Mm-hmm.
how scientists decide like which model organism are they going to use? Like, is it dependent on the if the organism shows more uh, like the like let's say we are researching about cancer. So how
Mm-hmm.
do we choose the model organism? Like which model organism should we use? Is that cancer is shown in that model organism which we are going to choose, or is it like just general?
Okay, so say for example, let me explain to you the general pipeline which is followed in research. So first of all, what type of cancer, what is your objective? Are you studying one new gene which is found to be, revolutionary in all types of cancers? Then what you would have to do, you would have to show its expression, its role, its physiological function, and its impact in all types of cancers using cell lines. That is the approach that is followed. First, you start with in vitro experiments, which is using cancer cell lines. So then you would have to take, say, for example, one breast cancer line, one glioblastoma line, all of these types of, right? Versus if you are trying to study the role of one gene in one type of cancer, then you go into a lot more detail. You've basically zoomed in. Oh, let me just switch on my video. Yeah, I'm just using expressions to show this. Yeah. So I was explaining, say, for example, if you're trying to characterize one particular gene in context of hormone dependent cancers. So in that case, you would take, say, breast cancer, you would take ovarian cancer. If something shows sex bias, it is only seen in male cancers, and accordingly, you use that for study. So all of that depends on your objective. Now coming to the general pipeline, first what you do, whatever hypothesis you have in mind, you have to investigate that using cell lines. That's the general approach. Um, there are some ifs and buts in this also. I'll come to that. So say for example, you have found gene X to be a huge driver of cancer. First, you will validate that in vitro. What you will do, you can do two types of experiments. One, you can take a normal cell line, normal as in these are called benign cell lines. So they are, I'll explain all of this as we progress, but just because you asked, let's go over it. Say, for example, you are working on breast cancer. So you have found some gene X and you're going to try and figure it out what it is doing in cancer. So you're going to take a normal or a benign breast cancer line and a benign breast cell line, and you're going to take a breast cancer cell line, both of them. What you will do, you will try to overexpress gene X in the benign cell line, and you will try to knock down or knock out that particular gene in the cancer cell line. What that will give you, it will give you a double check on the role of that gene in both contexts. So in a normal cell line, you expect it to lead to very aggressive disease. In the other one, you would, in the knockdown line or the knockout line, you will see decline in all of the cancerous properties. That will be your first level of validation. Once that is done, next what is done, traditionally done, is that you carry out the same validation in vivo. In vivo, usually in context of cancer, usually involves mice models. Why? A, because imagine the amount of research that is going on in cancer throughout the world. If you have, like, once you start learning a little bit more, you'll figure out so many research groups are working on it, right? So you would need a model which is easily accessible. You would need a model which is standardized across the world so that results become comparable. So therefore, what has been over time standardized by researchers is that they use nude or not skid mice. These are immunocompromised mice. I'll explain all of that in the upcoming slides. And what they do is whatever they have observed in vitro, they try to check the same thing in vivo as well. What is the role of it? It basically means that you're going to validate whatever observations or whatever conclusions have come up from cell lines. You're going to try and validate that when an organism is, you like, how do I explain it? When you're doing something in vitro using just cells, you are only using one type of cells. It's one culture dish which has, say, 5 million cells, and you are you're making whatever modifications to it, and you're studying what is happening in that monotypic culture. Versus when you are putting that cell population in an organism and then you are studying it, then you have the entire organism system. It has all of the possible physiological systems in place. It has a blood flow, it has oxygen. All of these different systems help you truly understand what happens around a tumor, because a tumor never grows in isolation, right? It is part of a system. So that is what has been standardized over time, that you use a immunocompromised mouse model and you validate your observations. Now, this is the general trend. What are the exceptions in it? Exceptions will first of all start off from the fact that where has your information come from? This I told you the most basic biology route which is followed till date, and it logically makes the most sense as well. If your information has come from patient data, like for example, if you have information from 500 patients and you're trying to check that, then in that case, only doing this much is not going to be enough. What you would also need to add to this pipeline, in vitro and in vivo pipeline, is that you would have to actually use uh, DNA, and it's not actually have to use, it's more logical to actually use patient and RNA, patient DNA, patient RNA, and extract protein from patient samples and validate that, okay, whatever expression you have seen from that data, you are able to see that in your context also. Say for example, you are using information from Australian cohort or American cohort, you would have to use patient data from India as well, and then you'd have to validate the same. So in this case, you are using in vitro system, which is your cell line. You are using clinical data. It actually increases the weightage of your study. Your journal uh, becomes more accepting of your data. You're strengthening the truth of the information that you're providing. So patient data or clinical data is something which is considered to be the most best confirmation you can provide for your hypothesis. Now, apart from that, in this case, in vivo validation would only go to mouse. You will not go beyond that. Now, where can there be other exceptions? I'll explain it to you when I talk about organoids in more detail. We'll come to that later. Some exceptions to this are also made in case of using Drosophila. Um, I'll give you one example because it's part of our department only. So somebody from another lab in our department, they work on Drosophila. So what was observed that if you have a certain gene known as ERG, a gene fusion between TMPRSS2 and ERG, I might have discussed it during the course. So it is found to be a major driver in context of prostate cancer. That gene, while it has been studied in mice and everything as well, that gene, very surprisingly, was found to give a very different phenotype in fruit flies as well. Like there was the lack of a particular wing. Like there's a default morphology of flies, and from that they could see a very distinctive phenotype that was lost when these particular mutation or this particular overexpression was present. So that entire study has been done using Drosophila, and that has been validated further using other models as well. So there are exceptions like this. If you can see a very clear, very distinctive phenotype in other animal models, then this route can also be chosen. But the traditional route uses, of course, in vitro cell line models. Nowadays, there are use of organoids, which is the intermediate preclinical model used for studies, and of course, in vivo validation. So that is the pipeline that is to be followed. If you ask me about C. elegans and Danio radio, honestly, it is debatable. It's C. elegans. It's not really very commonly used nowadays. It's like if you see here, the recapitulation of human physiology is extremely low. The difference between C. elegans and us is immense, especially when you have so many evolved models nowadays. It doesn't make sense to go that route anymore. Fish is still used till date. You will find a lot of labs in India who um, sort of work in that direction. In IIT Delhi, there's a professor, they all work in this domain. So they actually use, like I mentioned previously, they use the concept of regeneration to study a process. And they sometimes try to correlate with the idea of cancer. But yeah, that's not the traditional route for oncology research, not anymore. But for other types of research, you would need different models. Like, for example, rats are very commonly used, especially rats are used in rats or guinea pigs. These are used in when you're studying muscle biology, skeletal disorders, because um, guinea pigs are easier to observe. Like, for example, if you give them a task, they run around or they do some sort of exercise. Rats are used because rats are larger in size. So you have to, if you have to measure a parameter, like you measure their blood pressure, you measure their metabolic rate, etc., all of these things, then you need a slightly bigger model. Versus if your focus is entirely on some sort of neurobiological disorder, like if you're studying Alzheimer's, etc., then usually mouse or rat can be chosen. Sometimes rabbits are used. So it's usually depending on the type of disease, some standardized models are already used. But at the end of the day, if you are publishing a paper, if your data is logical and if it is supported based on actual data, if it's not a story that you're dictating to people, then anything is acceptable as long as it follows the science of it. Makes sense to a publisher, it makes sense to a journal and to panel of reviewers. It all is allowed. So yeah, it depends upon the context. But for our studies in cancer, in the field of cancer biology, you obviously have to check in cell lines in mouse as well. And yeah, PDX is something that will come about, and organoids are now the hotcake topic of study. So yeah, did you understand? Like, again, I can't give you a very definitive answer. I can only talk about cancer, but different models make more sense for different types of diseases, basically.
Yes, ma'am, I understood.
Okay. So, yeah, moving ahead, we'll first look at 2D models of cancer research. So, cell lines are in vitro model systems used in different kinds of field of medical research, like I mentioned, especially for basic cancer research and drug discovery. And their usefulness is primarily linked to their ability to provide an indefinite source of biological material for experimental purposes. This is very crucial. It's an in-depth, once you have a cell line, so people basically purchase, there are two ways of obtaining cell lines if you're setting up a lab. Either you form like a collaborative understanding with another lab who already owns it, who has generated that cell line and submitted it to public repository. You can have, there's something called MOU, which you, it's a sort of an agreement, or you can purchase it from something called as ATCC. I forgot to put it in the slide. Just know it for your reference. It's called American Type Cells Culture. I forgot the name. ATCC, American Type Cell Culture or something like this. So basically, they are a huge repository. They're a company, commercial repository, where they people submit their cell lines that they have all published and all established over decades, and they have these commercialized and they maintain them in culture, and they are able to provide it to you. So once you have purchased, you set up a lab. They're very expensive. You get one cell line for like lakhs and lakhs of rupees. You get it once, but then that is a resource that is going to last you forever. As long as you are publishing, as long as your next two generations are publishing, the cells are going to be fine. So that's one of their reasons why cell lines are, so extensively used. Having said that, the way I said that cells are going to be fine, there's a lot that has to be kept in mind. Um, Again, we go back to hallmarks, that senescence and all of those concepts that we discussed very early on about cancer cells. So what happens is that while cancer cells have become immortalized and they have, acquired the ability to keep on dividing without obeying Hayflick's limit and without having all of the other additional challenges that a normal cell has. Despite that, what happens is if you keep on passaging cells, that means if you keep on, if cells keep on dividing indefinitely, say, for example, I got a cell line in today in 2025, and then if I keep on allowing those cells to divide for the next 20 years, what is going to happen is eventually the cells are going to acquire random mutations, and the sanctity or the expected profile of that cell line is going to change. So, in order to maintain change in the sense that because they're cancer cells, they're going to acquire mutations anyway. For a cell line, what you expect, it is supposed to have a very predefined, very clear genomic profile. It is supposed to have XYZ mutations. It is supposed to have overexpression of XYZ genes, not have these expression. All of these things are very clearly profiled because they are standardized models for research. So to ensure that when you are continuously culturing cells in the lab and you don't indefinitely keep on culturing them more and more in number and they don't change from their original profile completely, a few things have to be kept in mind. Like, for example, what you do, that if you're starting off, say, for example, I'm acquiring cells today in 2025, then my first job would be to expand those cells in number, of course, so that I have like 50 million cells. I will get, say, for example, 2 million cells when I purchase it from the company from HECC. I have to expand it in number. Then what am I supposed to do? I'm supposed to have stores. I'm supposed to have backups of that particular cell line. So for that, cells are basically frozen down. So if you have done cell culture, you'll be familiar with it. So you preserve those cells and they stay fine for very long durations of time. The rest, what you do, you allow that to stay in culture. Um, if you don't know how cells are frozen, basically mammalian cells under normal circumstances are frozen in FBS, which is fetal bovine serum, along with DMSO, which is dimethyl sulfoxide, and this acts as a cryoprotectant so that when you are freezing cells in these very ultra low temperatures, they don't form ice and they don't die because, like, the cells don't die because of mechanical damage. So that's how cells are preserved. So what you will now have, you'll have a repository, and this repository is stored in ultra low temperatures in minus 196 within liquid nitrogen, and they stay fine for like years, for decades. They can stay fine for 10, 12 years easily. And the rest you keep on passaging. Simultaneously, what is also done, cells are every few years, every like 4 to 5 years or so, it is recommended that you subject the cells to something called as STR profiling, which is short tandem repeat profiling. So what that allows you to do, there are companies that actually have this. It's a service provided by a lot of companies. So you extract the genomic DNA from these cells, send it to the company, and what they will do is they will perform complete sequencing of that particular cell line, and especially they'll sequence the short tandem repeats, and they will validate that okay, whatever was the original profile versus what is the current profile, and they'll match. You will expect some deviation to be present because cells have divided over time, but it should not be so huge that the cell profile has changed completely. So commercially, this is done. It's a prerequisite of a lot of journals before publishing. So this is something standardized over time in labs so that people know that, okay, the cells that I have received are the cells that is actually being used by other labs. And, you basically, it's a cross-check validation of your cell lines. Okay, so coming back here. So this has become your indefinite source of biological material. So the question is, how are these cell lines established in the first go? So they are established by isolating cancer cells from patient-specific organs, like the organ where cancer has arisen, and then growing it in artificial culture media, which when you transplant into immunocompetent mice or immunodeficient, in this case, it has been written as immunocompetent. It actually should be more commonly, it should be immunodeficient. It leads to the development of cancer cell line xenografts. So, crux of the story, basically, usually cell lines, like when they were standardized, they were established from people who a had quite like, how do I explain it, different types of cell lines are present. Say for example, if a disease, if any cancer has 20 subtypes, like 20 subtypes mean 20 types of mutations, 20 types of alterations overall, then it would be ideal to have a representative model of each of these subtypes, right? Now, while that sounds like a very idealistic situation, it's very difficult to actually do because you would need to have a patient who agrees to donate their cells for this purpose, and then you would have to standardize the protocol for establishment of that cell line. So, just out of curiosity, just if you have time on hand, go to the website of ATCC and look at any cell line, whatever is interesting to you, whatever cancer you are interested in, and you will find all of these different names. Like, for example, if you go to prostate cancer cell lines, um, take for example, there are some cell lines called as 22RV1, VCAP, all of these cell lines. If you open the details of it, you will get all the information. Who was the patient? Like the name of the patient is not given, but how old was the patient? Where was the patient from? Like the race of the patient, because like I explained previously, depending on where a cancer has arisen, you would have different phenotypes. Then their age, if they have any other condition, if they had metastasis and other portions of the body, if the cancer has been established from the primary tumor or from the metastasized tumor, because depending on that, the cells would have different properties. A primary cell tumor is less aggressive versus something derived from brain metastasis would be more aggressive, or lung metastasis would be more aggressive. So all of that information is provided. So basically, what happens then is that you have to like you extract basically those cells from the patient, and then you culture it basically means um, you supplement the cells with a medium which is enriched in some specific reagents which help in cell division, which help in preventing apoptosis. There are a lot of reagents which are available for it. There are some standardized culture media also, like RPMI and DMEM, if you're familiar with them. These are the most commonly used mammalian cell culture media. So all of that is utilized, and that helps in controlling cell cycle and regulating cell death. And cells are provided these optimal conditions, and then they can be repeatedly passaged to establish a model cell line. So if you look at the steps one by one, you have, if you were to establish a cell line yourself, how would you do so? You get your sample. Say, for example, you receive a breast cancer sample, and first you have to dissociate the tumor cells because they are all part of a tissue, so they would all be adherent to each other. So you use enzymes to separate those, and then you use that dissociated cell and you culture it, or you allow it to grow in the culture media, and you expand them over time. Like, for example, here it has been mentioned 8 weeks, but if you have to commercialize a cell line, you have to do it for much longer. And then you can establish eventually a viable cell line, which can be used for cryopreservation. So, this is the broad idea. So, the example that I'm giving you right now is a very um, like one of the main discoveries and one of the biggest bioethical disputes in the field of cancer biology, not even cancer biology, disease biology in general. So in 1951, Dr. George Otto Gay, he was from Johns Hopkins Hospital in Baltimore, and he established the first cancer cell line called as HeLa. You must have heard of it at some point of time. So yeah, see here, it is a picture taken from ATCC. So it is called as the HeLa cell line, and it is a cervical cancer cell line. So this was the first example. This is how it looks. If you zoom in, you'll be able to see how the cell looks. Again, keep in mind, this is just a representation of one type of cell line. Each cell line looks different. And one of the major features that I would like you to observe very clearly is that if you just look at this this dotted sort of a structure, I don't think it will zoom further. But yeah, look at the tiny little dots. They are all representatives of nucleoli present within nucleus. So that indicates how rapidly transcription and translation, etc., is taking place yeah, within these cells. So that is something you can very easily see under the microscope. Now, because HeLa cells are so large in size, that's why they are much more easier to visualize. But if cells are small, it's just more difficult to see. But yeah, this is one example, and this is the woman who is attributed with the generation of this cell line. Her name is Henrietta Lacks. And I would really urge you to go and read up on the bioethical dispute, which is associated with this cell line. There are two sides of this story, but just to cut the long story short, basically this, according to popular history, it is considered that her family, or rather Henrietta Lacks herself, was not informed when her tissue was being used for establishment of the cell line, and that goes completely against the bioethical norms, and therefore there were lots of claims made by her family, and therefore there was a lot of dispute about whether or not a biological material from a certain person can be utilized without their permission or without their prior consent. So that has led to the generation of a whole set of laws which have come in about prior informed consent for patients. So any individual in today's time, if they are to work with any patient tissue, there has to be an agreement first of all between the researcher and the source of that patient tissue. Usually, of course, that has to be a medical facility, a hospital of some sort. And not just that, the apart from the legal agreement between both of these administrative groups, each patient or at least their family member has to be informed, and there has to be a written record of the fact that they have agreed to contribute their um, a part of their body for research. One of the major reasons is because it's something, as you can understand, once somebody has your cells without your consent, anything can be done with it, right? Your living cells, they can be used for profiling, they can be modified. Your genetic information is now for public use. Kallo, kallo, Rams. So it can be used for public use. And therefore, the HeLa cell line was something that was done. Obviously, understand that it was the first major discovery, and therefore there was a lot of laws that were very gray, and so it's a very interesting story. Give it a read sometime. It got partially resolved only a few years ago, so it was a rather recent event where the family, I think, was not awarded any money, as far as I remember, but read up on it. Another example is of Dr. Robert James Valentine. He established the Raji cell line, which was derived from a patient who was affected by Burkitt's lymphoma, and it was the first human continuous hematopoietic cell line. So that was another example of an established cell line in recent time, not recent times, but one of the first cell lines to be established. Now, one thing to keep in mind, like I've already mentioned, is that the reason why cell lines, apart from the fact that they are a lifetime resource, one of their major advantages is the fact that the alterations present within that patient will now be available as a model system for anybody who wants to research that subtype of the disease. So, copy number alterations or even the transcriptional profile of these cell lines would show alterations similar to that which you see in the primary human tumor, which you actually see in that patient. Okay, so that's why their relevance is, of course, immense, and their usability is huge. But despite these advantages, like we have discussed, there are a lot of limitations to their usage as well. Like, first of all, the lack of heterogeneity. Like I mentioned, since you have cells which have been immortalized and stabilized over generations, what happens is you have a homotypic culture, one type of cells just increasing more and more and more and more in number. So what happens is that over several generations, they will start to lose any heterogeneity, or they will not have any heterogeneity for that matter, or the multifocality. Multifocality means that certain cancers, they grow as foci. Um, like for example, again, prostate cancer, for example, it grows in a like um, how do I explain? Think of a large tumor. It has smaller subsections. The disease, by its inherent nature, is such that smaller sections have different types of genetic alterations which lead to eventual formation of the disease. So, therefore, if you take one chunk out of the entire tumor, then that is not going to show you all of the possible diversity within that entire tumor or within that entire cancer. So, you're getting partial information. The same thing happens with use of cell lines as well, that they completely lose the diversity or the heterogeneity within the disease, and it becomes very monodirectional in nature. That's why you need patient derived. That's the concept that has emerged over times, that people are trying to A, look at 3D models, B, look at patient information, because that is going to give you true information, truly preclinical information. Then the limited representation is a major challenge. Like several tumor subtypes are absent. Like I just mentioned, that if you have 20 different subtypes, it would be ideal to have representative models of 20 different subtypes. But it's not that easy. It doesn't even exist. For most cancers, you don't have representative models of every type of disease. And so a lot of it goes unrepresented. Examples being luminal A breast cancer subtype is not found in the cell line panel, or the exocrine like subtype of pancreatic ductal adenocarcinoma is not available in the cell line panel. So these are just some examples of the same. Next, the lack of stromal compartments, which means that you're missing out on the other contributors when you talk about cell lines, like I just explained, because they are one type of cells growing in a layer. You do not have the contributions from lymphatic vessels, from the ECM which surrounds it, from associated immune cells, from blood, from fibroblast. None of that is present. All you have are the cancer cells. So it is not representing the condition of a tumor in vitro. It is just a preliminary study model. Furthermore, the adherent two-dimensional nature of cell growth is also a limiting factor. That is because cells in a tumor never grow like this, right? They're always forming some sort of a mass. So cells on the outside versus cells which are at the core of the tumor have very different exposures. They have very different influence from the surrounding environment. And based on that, a lot of things can differ. Okay. So how do you tackle this problem? You move towards 3D models for cancer research. So these are two representative images. You will find mind-blowing images if you go into this. These are examples of some organoids. I think this one was a cervical cancer patient organoid. This was a brain, so a brain, yeah, it was a brain organoid, cerebral organoid, basically. Yeah. So these are two examples of organoids. So like you can see, these are huge round structures. What you see on the inside, these small, small ones, they are little cells. And you can see how within each cell, I think this was staining for some sort of a protein. I don't recollect it, but probably some nuclear protein which is present. That's why you can see, and you can see that it is not continuous. There are some areas which are hollow, some areas which are less hollow. Same thing for cerebral organoids as well. And also, what you need to see is that not every organoid looks the same. They have different morphologies, and that depends upon the type of cancer. They do not look like a complete, beautiful, round structure. That's not how it works. Certain types of organoids grow in certain ways. That's just the way it is. Okay, so a quick historical perspective, quick because yeah, we're running a little slow, but yeah, it's okay. We have enough time to wrap it up. So just a timeline perspective of the whole thing. So the first human cell line, HeLa, established in 1951. Yeah, you can refer to these PMIDs also. These are basically the PubMed IDs for you to go into and just give it a read if it is interesting to you, if you want to write a review on something like this. So it would be a wonderful way to study. Then, first, colon cancer patient-derived xenograft model, which is a PDX model. We'll come to it. Then, the genetically engineered mouse mammary cancer model. Then, the formation of a, oh, sorry, yeah, this came first. Multicellular spheroid cultures. Then came the mouse memory model. Then the colon carcinoma cell line is grown in transwells. Transwell is like a um, how do I explain? Think of it like a small cone, basically like a small cone. Think of it as a small container where the base of it is a very, very fine mesh. And that mesh is the reason it's called a transwell because it is something which allows the movement of cells, but in a very restricted, very controlled manner. And transwells are very commonly used for studying invasion and migration capability of cells. It's extensively done. We do it in our lab, and there are other ways to do it also. But transwell insert mediated migration and invasion of cancer cells is studied. So it's you can look it up. You'll understand. It basically that mesh serves as a membrane across which cells will be able to move. So that's the idea of replicating the invasive and migratory potential of cells. Then you have the development of a lung on a chip microfluidic device. This is something beyond the scope of this course, but just to briefly explain it to you, there are some sort of think of them as little slides. And also, let me be very clear, I'm not an expert on this, but whatever literature I have studied and I know, based on that, think of a slide or a panel, and that panel has a little caveat. Okay, that caveat is going to have the representation of a blood flow, and lining that caveat will be a mass of tumor cells. What is the logic behind it? The logic behind it is that when cells are undergoing metastasis and they're moving through the blood or they're moving through the lymph, they are exposed to some mechanical pressure as well, right? Because the blood is flowing and they are moving in blood. So there's a lot of force that is coming in. There are other cells that are coming in. So the sort of factor in vasculature, which means the flow, which is affecting the flux, which is affecting the movement of cells. These devices have also been changed, like they have been developed over time. So lung on a chip is just an example. Organ on a chip is the basic concept. So you try to replicate the movement which is seen in the organ and which is faced by tumor cells when they are within that organ. You try to duplicate that on a slide. So it's like a chota sa chamber, and through that chamber you allow the fluid to pass through, and then you try to monitor the impact of that on these cells, and different types of parameters are measured. But yeah, it's a very, very cutting edge way of studying. So if it is interesting to you, it's a wonderful area, has a lot of funding. So if it's interesting to you, you can definitely look at that. And there are, while it's not that extensively done in India, if you want to go into research in this domain of organ on a chip, etc., you can look at a lot of labs. Of course, in USA, you'll find a lot of labs. In Germany, you'll find a lot of labs. In Iran, surprisingly, you'll find a lot of labs that actually work in this entire domain. So they have great funding, especially in context of brain organoids, in lung organoids, all of that. So if it's interesting to you, read about it, and it will stay relevant at least for the next few decades. So you can explore that direction if it's interesting to you. Um, then came the generation of the first colon adenocarcinoma PDO, which is patient-derived organoid, the microfluidic model of a tumor microenvironment on a chip. So people have started now addressing the fact that because cell lines are limiting in context of having only one type of cells, they have tried co-culturing cells, co-culturing immune cells along with your cancer cells. So cells which are present around a tumor in the tumor microenvironment, they have started co-culturing that and seeing how the cancer cell and the microenvironment they interact with each other. So that is also being done on a chip. So that is how you model the tumor microenvironment on a chip. Again, as you can see, relatively very, very recent technique. It's like it might not look very recent to you, but if you think about research as a whole, 15 years is really not that long a time. And This is something which is still which is meant to be used as a study platform. It's not meant to be, not at least not immediately meant to be something used for patients, but yeah, it's a model of study. Then the air-liquid interface for gastrointestinal organoids is being studied. The organoid modeling of the TME or the tumor microenvironment is being done. Multi-compartment tumor model on a chip is being done. Microfluidic platforms for tumor organoids in 2020 have been automated so that, of course, you try to make this high throughput as well. That is, well, in a blunt way to put it, that's how you make money. So you make things automated, you allow people to use that as a service, and you try to study different types of cancers on microfluidic platforms. Of course, depends on what type of cancer it is and how relevant it is. But yeah, this is being done, and it's just to get you abreast with how the world is working, how academia is working, because this is not stuff you find in books. You don't get to learn about all of this. So just to introduce you to the idea what the world is chasing right now and where it will be 15, 20 years from now. So read about it, figure out what is interesting to you, and then you can explore that in the long term. But for now, what we'll focus on is what has been sort of more well understood. So, what is better characterized, and that is the direction we're going to take. So, when we talk about 3D patient derived models, these are primarily the subtypes which are looked at. First, of course, what we have already discussed, this is not a 3D model, but this is a patient derived model. So, we have included it under the same umbrella. So these are 2D cell lines, which of course are easy to maintain. There's a lot of versatility which you can do, low cost, and of course, high reproducibility. So a million people across the globe, if they're using the same cell line, they are expected to get the same result with the same experiment. So that validates the sanctity of your finding. So that's why it is still a model that has not gone out of fashion, and you can expect it not to go out of fashion anytime soon. However, it is less biologically relevant in today's time, considering how much has changed in the last decade and a half. Second, it does not factor in the microenvironment, like we've already discussed. It has unnatural adhesion forces, which means the cells are very closely adherent with each other. No cellular heterogeneity. Already we have discussed this as well, that only one type of cells are growing in culture. Second, what I just mentioned, the organ on a chip, you have, yeah, this is basically what it looks like. You have different types of chips depending on what type of example is being taken into consideration. But yeah, you are um, duplicating the tissue-tissue interface. You can co-culture cells. You will have a controlled environment. But yeah, while it is cutting edge, it has very confusing disadvantages as well. Like, for example, the same chip cannot always be duplicated. The same lab might make different types of chips. But yeah, it is actually in the domain of mechanobiology, where I am not an expert at. So I'm not going to be very helpful in this regard. I'm just trying to introduce you to this concept. If it's interesting, you can look at it. I can come back with more information later if you want it. But yeah, these are there's a lot of inconsistency that can often be seen. It's difficult to duplicate that result, and protocols cannot always be standardized. It's usually the intellectual property of one lab, and then you can outsource your cell lines or your project of interest to that particular lab, and then they can run experiments for you. So while collaboratively it is possible, it's not always the route that somebody wants to choose. It's easier to have a commercially viable chip, and then you can just purchase it and do your experiments. So yeah, this is a slightly dicey field that has a lot of open ends. So that's why we are trying to steer clear of it. Let's give it a few more years, and then the discussion will be more concrete in this regard. Then we come to the concept of spheroids. So spheroid, and then another term is organoid. So first, you must understand the difference between the two. Spheroid, as the name literally means, it is a sphere. So you have these patient-derived cells, and you give them a particular condition whereby um, they can form spherical, like, because if you give them limiting conditions where they don't become adherent in nature, they will form these balls, these clump-like balls, and they would be derived from one type of patient. So for spheroids, actually can also be generated from cell lines as well. If you deprive cell lines of certain conditions, and if you supplement them with a particular kind of media, like with EGF, with transferrin, insulin, etc., there are different compositions which are required. Then cells, instead of being adhered to a surface, and if you give them a non-coated culture dish, they will start forming these little balls, which are called as spheroids or tumor spheres, as more commonly they are known. And that ability is also often measured in a lot of experiments. So the ability of a cell or of a population of cells to form a tumor is usually considered to be a better um, to be a pro-cancerous advantage, and therefore if a certain condition is compromising that ability of a cell, it basically inhibits their stemness capability. They have less stem cell like feature, and therefore that would be a anti-cancerous strategy, and it can potentially be used ahead. So, spheroids, they have poor structural organization. They do not get a lot of oxygen to the core, and spheroid size can be very variable. And they are not they don't actually represent the genetic heterogeneity of the patient. However, organoids, they actually contain lots of cell lineages. They are derived from many different cell types, which are taken from a particular organ. Sorry. So, for example, say for example, if you're taking up, I'll give you an example of prostate cancer because that's what I work on. So what I get is I get a sample of the tissue from the patient, from a patient who is undergoing biopsy basically. So from that sample, I have to culture those cells, and I culture them in a manner which only selects for stem cells, or which preferentially select for stem cells. And what it does is it enriches that population, while the other cells which do not have the stemness capacity will naturally die because the media is not supporting the growth of non-stem cells. Then what happens? They start taking up their default shape, and prostate cancer by virtue forms sort of a circular structure only, but a lot of different organoids have very different structures. Like, for example, if you remember the image I showed you, this is a cervical cancer organoid, so that looks pretty much circular, it's by itself only. But if you look at the brain organoid, you can see that it has this very strange shape. I don't even know what to call it, but it has this irregular sort of morphology. So then all of these cells, they have assembled and they have taken up a certain shape. And what you can see small these sort of caveats which are also present, which have which is unique to that type of model system. And it is what it is effectively doing is it is duplicating how the tumor formation actually occurs in that organ in that patient. And it can have cells of different lineages also. So different types of cells can get selected, like epithelial cells, basal cells, within epithelial cells different subtypes. Some sort of organoid cultures even allow co-culture with immune cells, so you will have that as well. This is all very cutting edge. It's eventually developing. There's endless information to get there, but for your understanding, what you need to understand is that patient organ derived cells, when they are cultured in the appropriate medium, what they will eventually do is they will form these structures that will form, that you will call as an organoid, and they will actually recapitulate the histology, the organization, the genetic variability which is present in the patient. So a brain derived organoid, or a brain patient derived organoid, if you section it and if you stain it, you will see that the way in which the cells are organized, that is supposed to be very similar to what you see in a patient, in the patient where it was derived from. The mutations that you see in this particular organoid should recapitulate what you saw in that patient. So all of these things, what it effectively help you confirm is that now you have a model system which is an exact duplicate of that patient. So whatever studies you do, you can actually you can make it happen. You can facilitate it in the organoid, and then that will potentially work very well for that particular patient, or for a group of patients who have the similar type or similar subtype of disease. So you now have a new model which is very precise, which is very individual specific, but that has very far reaching potential. Okay. So that is the idea. You have patient specific organoids. You can, however, there are challenges with every technique, as you can understand at this point. First of all, interpatient variability can be huge. Like, not everything will be very easily applicable, but despite all of the challenges, it is still a huge step ahead from using cell lines where you just took one type of cell and now you are actually trying to recapitulate what is present in a patient in your lab. That's a huge, huge thing to achieve. Second, the downstream analysis is immensely expensive. More than that, the establishment itself is very, very expensive. As you can understand, you are giving very controlled media conditions. You cannot use a generic media which is used for culturing cell lines because they are a lot more robust. Here you are using patient cells. Of course, safety is also something that needs to be considered. You need to make sure that patient is not like HIV positive or they do not carry any other very serious infections. All of that has to be kept in mind. Establishment cost is very high. Success rate in a lot of cases is very low. Like if you have 10 samples, most of the time, hardly two or three samples of them can actually form organoids. However, your effort and your investment goes into all of them. So while it sounds amazing, it's very difficult to actually make it happen. And finally, rodent xenografts are sorry, rodent xenografts. Rodents basically mean mice model. Mice model xenografts are also used. They are very accurate in terms of replicating tumor genetics. Sometimes you can manipulate the patient, manipulate in the sense you can change or alter the genetic background of the tumor prior to implantation. It factors in the complex microenvironment associated with the disease. But what is its problem? First of all, you are injecting human cells or implanting human cells in a non-human animal, right? It's a non-human environment. So the immune system is completely different. Secondly, because this is a challenge, what is actually used in most cases is not an immunocompetent model, but instead an immunocompromised model. Because otherwise, it is often very difficult to allow those cells to grow at all. So there are debates about this as well. Secondly, resource is immense. As you can imagine, maintaining a repository of such animals is very cost, like the cost is very huge, and not just cost, it's also the reagents which are required to actually sustain such a colony for a very long term. But also, before you can claim anything in the field of, you know, medical oncology, all of this has to be done because, as you can understand, you are adding to known literature and you're claiming that whatever finding you have made is an advancement in the field of a disease which is killing millions of people every year. So the responsibility that comes with it is of course immense, and so all of these different stages of validation have been put in even before people, like when I say people I mean clinical trials, can take something ahead for approval. So preclinical model verification needs to be done, doctors need to be involved, and a lot of validation has to be done before anything is even taken to clinics and patients are enrolled, and then the drug panel is investigated, and after going multiple rounds of clinical trials, at least 3 to 4 rounds, which take at least a period of 5 6 years, only then whatever you have discovered can actually be translated. So the concept of bench to bedside sounds very idealistic, but there's a lot of verification that occurs along the way. So, yeah, these are all very cost intensive topics that I've told you about. So, this is just a quick timeline recap of organoids so that you get an idea about how far advancements are actually being made. And as you can see, first people learn about the fact that cells can self-organize and regenerate in as early as 1907. The first incident of an of using patient tissues or trying to manipulate cells into behaving a certain way, that came from the discovery made by these two amazing scientists, John Gurdon and Shinya Yamanaka, for their most outstanding contribution in the field of medical biology, especially in the field of developmental biology, which is the idea of human iPSCs. So, you might have heard of this name if you have studied developmental biology, Yamanaka factors. So, he was the scientist who is credited with that discovery. So, they basically figured out the strategy which can be used to manipulate cells and generate induced pluripotent stem cells. So basically, you can reprogram an adult cell, like for example, a cell from my body, a somatic cell from my body, and it can be modified in a way that it acquires a stemness property. And because of that acquisition from an adult stem cell, it can generate any possible stem cell of that lineage. For example, if say somebody is able to extract some tissue from my lung, then they would be able to reprogram it and then generate an entire lung organoid. If it has that stemness potential, of course, they are potentially capable of generating an entire organoid, which is representing all of the different types of cells in my lung, and they would be able to recapitulate it. So that was only possible because of this first discovery that was made by them. And over time, of course, a lot has evolved, a lot of changes, a lot of commercial changes have been made, and now standardization of techniques has happened. We take all of that into factor, but that first discovery was in 2006, and it's a huge contribution, and it is something you should definitely know about, not just for exams, but because you hopefully aspire to be a scientist in the long run. So after this discovery, one of the first types of organoids that were to be made was of the cerebral cortex. Then people have established intestinal organoids, retina-based organoids, kidney organoids, prostate organoids, pancreatic cancer organoids, cardiac organoids, uh, fetal liver-derived organoid, and this was, I think, the most recent, which was a testis organoid, based on this publication. But after that, I think I read about placental organoids also, that people have modeled the placenta. I mean, imagine how much science has progressed, and such unbelievable discoveries are being made by people that they are basically uh, generating a replica of the placenta in their labs. So a lot of women who would be, can potentially suffer from a lot of diseases during their gestational period, or possibly some diseases which can influence the growth or the development of neonatals. So all of that can be studied, all of that can be modeled, and if necessarily, if possible, if somebody suffers a rupture from their in their placenta, then there can be generation of placental tissue in the lab, and then that can be used to help the patient. So much can be done. Read about it. Like I think I read about something similar being attempted, but I'm not sure. But yeah, read about it if it is interesting to you. Read about it. People are doing such incredible work across the world. So these were some of the most interesting discoveries that I wanted to just introduce to you. I think this is our last slide for today. So, all of the things that I discussed till now, they are still considered to be in vitro. Why in vitro? Because whatever we have discussed today, they have all been a part of models that you can actually culture and study in the lab. Despite the fact that it might be a cell line, it might be patient derived, it is still cultured in the lab, so it is in vitro in nature. And what I had discussed initially is in vivo, that is a separate thing. In context of oncology research, whenever, like I mentioned initially also, whenever we talk about in vivo models, we think about mice. So one of the reasons why mice are almost inevitable in today's time is because of this particular issue, is that for actually studying how effective a drug is going to be against a certain type of cancer, validation needs to be done in vivo, because the rate at which a drug can be taken up by adherent cancer cells versus the rate at which that drug can be taken up by a clump of cancer cells who have formed a tumor, that is going to be different. Cells in the center will not even be exposed to that drug, right? So the rate of uptake varies, and therefore the behavior of cancer cells in its response is going to be different. This is just one example, but there are many other advantages and disadvantages as well. So, over time, this cancer drug development domain has been affected by the absence of any preclinical cancer models that can actually evaluate the clinical testing of novel compounds in human patients. So, to overcome that, people have utilized patient-derived tumor xenograft in immunocompetent mice, which are preclinical models. So, for that, they have actually used basically, in that case, what is done, instead of generating an organoid in the lab, people take the tumor tissue and process it a little bit, and then they implant it into the mouse directly. And then in the mouse, those cells they form a xenograft, and so it is a patient-derived xenograft. I hope you know what a xenograft is. It's xeno basically means that the species of origin is different and the host species is different. So that is a xenograft. Also read about autograft and allograft, the differences between those. So basically, people have tried to do that, and then they have tried to figure out how the drug, how effective the drug is in these PDXs or patient-derived xenografts in that case to use as a preclinical model. And the type of mouse that is used in such cases include either nude mice, I already introduced it to you some time back, then these severe combined immunodeficiency mice, which are SCID mice, non-obese diabetic or non-SCID gamma mice. These are severely immunocompromised mice, and RAG and non-RAG gamma mice. So I'll go over this briefly, but it's going to be a little redundant, so we'll go over it quickly. Basically, what you need to understand is each of these different mice, they have different strains. What do I mean by strains? These are also commercially available. Majorly, you can look at two websites if you want to learn more about it. One is Charles River, and yeah, maybe look at Charles River only for your understanding. But yeah, technically you can look at uh, any concept for your understanding. But Jackson Labs and Charles River's labs, these are the two major commercial hubs which supply mice models to most of these labs who are working on cancers in general. So these different types of mice, they have very specific strains. The strain basically means in this mouse model or in this colony of mouse models, they will all have the same set of mutations, which is rendering that immunodeficient phenotype. So, for example, let's go over them one by one. Let's look at nude mice. Nude mice were the first immunodeficient mouse strain. They have total number of circulating lymphocytes almost 5 to 6 times less compared to a normal mouse. Majority of those cells are B cells, so they're used for cancer metabolomic research extensively. However, they also have high correct prediction rates and compared to in comparison to in vitro systems for tumor sensitivity and for studying tumor resistance. So they seem like very good models for study. However, but the limitation of testing is very, very less. At least 4 months are required for rapidly growing tumors and 2 years for slow growing tumors so that you can actually run your experiment. Secondly, they are very expensive, so they need special conditions to avoid infections. Keep in mind, any immunodeficient mouse model needs a lot of caution when you are handling the mouse, because any infection that you carry with yourself while you're handling the mouse can also affect them, and they don't have an immune system, so they're not going to make it. So you need to be very careful with that. Then comes the next subtype, yeah, which is the skid mice, which have the severe combined immunodeficiency syndrome phenotype. They don't have mature B or T cells, and they have decreased NK cell activity as well. They provide realistic heterogeneity of tumor cells. They can predict the response to a drug, and they can allow the rapid analysis of tumor response to a therapeutic regime. Uh, because they are immunocompromised, at the same time, they do not actually duplicate a tumor microenvironment because there is no immune microenvironment present at all. But at the same time, they also allow for very rapid growth of tumors, so you can actually run your experiment a lot more smoothly. Low level of engraftment of human cells, and they have a very short lifespan, approximately of 8.5 months, maximum 12 months. Beyond that, they start acquiring mutations of their own. They become very old, and more often than not, they'll die naturally. So that's what happens. Then you have NSG mice, easy to prepare, they live longer. They don't have this IL-2 receptor gamma chain, and they lack BG, NK cells, and also cytokine signaling. Sorry, cytokine signaling is absent. They can be used for human metabolomic studies. They're very expensive. They're severely immunocompromised, so they acquire diseases this fast. They are very sensitive. So yeah, that's a major problem. Then rag mice have very similar mutations to skid mice, but they also carry these rag1, rag2 mutations. If you have studied immunology, you would know that rag1 and rag2 are the recombinase activating genes, which are necessary for generating B cell and T cell diversity and formation of more and more, not B cell T cell diversity, BCR TCR diversity, which means antibody generation and T cell receptor generation process. So in that case, also it has its fair share of advantages. But the NRG mice possesses this RAG1 and RAG1 and IL-2 are gamma chain mutation. They are better. They have better tolerance to irradiation, and they can provide easy access for implantation as well. So they have their own advantages, but uh, they have better implantation in the newborn. But then you can't always use the newborn because it depends upon what is the stage of onset of a disease. Like, for example, if your cancer is more specific to adults, if there's an age factor associated with it, then you need to use slightly older mice, like 6 weeks, 8 week old mice. So they might not have that good an engraftment level. Or sometimes the xenospecific selection of human T cells might occur. So that is also a huge disadvantage. So these are the different properties that you can see. But if you want to learn about the genetic background that is different between these cell types. Uh, I'll show that to you. Let me just wrap up the next slide. Then I'll show it to you how to figure this out. Um, Let me wrap it up quickly. Basically, oncology research, it involves extensive use of model systems to study disease progression and for molecular characterization. Cell lines are used till date, but they lack heterogeneity and their adherent nature limits their use. Recent advances have focused on the use of 3D systems for this purpose instead. Despite their wide range of applications, patient-derived models also have their own set of challenges. Final validation largely involves immunocompetent or immunocompromised mice models, depending upon what type of study you're performing. What you should look into is that what exactly are these limitations of PDOs or patient-derived models for that matter. Did not go into much detail about it, so just explore it a little bit. Um, and why are they not so extensively used? Why are cell lines still the standard of study? Why is patient-derived model not the norm? And which model system would you suggest? Which would you think is the most ideal model system for performing in vivo studies and oncology research? So think about it. This might be an interesting angle to look at. So that basically sort of wraps up our class for today. But I want to show you something. So if you want to look at the different genetic backgrounds of all of the different models which are used, the immunocompromised mice models. So go to Jackson Labs. Yeah, so this is the Jackson Lab. You would find, okay, let's just look at node. Let me pick this one. Yeah, oops. I think there might be some issue with it. Just give me a second. Yeah, so this is the other one I was talking about, Charles Rivers Lab. Uh, Not Skid Mice, Jackson Lab. You can look at any of them. I'm just looking at Jackson Lab because that's what I use regularly, so I'm more familiar with it. So if you go into details about any of these mice, this is what you will see, right? This is the strain of the mouse. The common name is written like this, not skid or not skid like this. This is the strain number and this is the background. If you go into more details, you would figure out how this mouse was generated. So more and more details are mentioned. Like for example, if you look at this particular strain, these are they carry the SCID mutation in this PRKDC gene, as a result of which they lack these functional BT cells and they have all these different changes. They carry the SCID mice, they carry a DNA repair defect so that the genes which code for antigen specific receptors on lymphocytes, that is no longer able to happen, and therefore they don't have a functional immune system. So this is one example. The development process, you can figure out all of these details, like which mice had to be crossed, because the more complex the genetic background of a mouse, the more number of crosses it must have undergone between mice of different backgrounds as well. So you'll find all of these differences, the significance of each allele. You'll be able to figure out what type of gene it is, what is its role, all of that can be used. The way in which these mice are supposed to grow, or the expected growth of these mice, can be figured out. Like obviously for mice, age is in weeks, and that there's a conversion rate between the age of a mouse and the age of a human. So accordingly, it is also correlated. So all of that information you can figure out. Um, there are control animals as well. Yeah, this is how they look. Very tiny, where they're like the small, very tiny mice. The adults are slightly bigger, like 20, 25 grams usually, but the small ones are like really tiny, the baby mice. So this is just one example of the not-skinned mice. The NSG mice, you can read about it. And this is a commercial name. The background is different, but you will find similar immunodeficient mice models from many different companies. So you can refer to whichever is more comfortable for you, or whichever is more commonly procured in the lab that you worked in or want to work in. So all of that is just accessible information over the internet. So yeah, this is basically just what I wanted to show you. And yeah, I think I am done with the information I wanted to share for today's class. If there are any doubts, please go ahead and ask me, and then we'll wrap up today's session.
No, ma'am.
Okay.
Ma'am, uh,
Yeah.
first question, the limitation of patient-derived model. Can
Mhm.
we say that it must be a immunocompromised, therefore, in chances of infection was very uh, fortunate about infection?
No. The infection thing we talked about was for mouse models. This
Okay.
we are talking about patient-derived models like organoids or spheroids or the
Okay.
organ on chip, all of those limitations
Okay.
of those. The
Okay.
immunocompromised mice are extensively used. You cannot do cancer research without it. I mean, you can do it, it will just not be acceptable for journals. So yeah, this is actually more just think about it. It is it's going to be a technical and a logical challenge towards their extensive usage. So think about it. You'll find answers online to read about it a little bit.
Thank you, ma'am.
Anything else? No problem. Anything else? Okay, if not, then thank you so much for joining. We'll have the next class again on next Monday, and we have three more classes left. So next class will probably discuss the remaining parts of precision medicine, and then we'll move into those terminologies that I wanted to discuss with you all about hazard ratios and all of those terms about biochemical recurrence, etc. And in the last class, second last class, we'll discuss drug resistance and tumor evolution to some extent. And the final class on 31st will be a sort of a wrap up class from the entire course. So that should sum up the entire course. So yeah, I'll see you on Monday. Thank you so much for joining. Have a good night.
Thank you, ma'am. Good evening.